So today we're going to be hearing from Dr. Joost Lesterhuis. And I have to just have my notes up because the microphone's really sensitive today. So Dr. Lesterhuis is an NHMRC R.D. Wright Research Fellow at the University of Western Australia and an Honorary Research Fellow at Telethon Kids Institute. He is a medical oncologist and a medical scientist researcher in tumour immunology and cancer biology, and it's a really rare combination of qualifications to have someone with both a medical qualification and a medical scientist as well. His research focuses on identifying new effective treatment combinations in mesothelioma and sarcoma that focus on immunotherapy. And for those of you who are update regulars, you'll have heard Michael Millward's update on immunotherapy maybe a year or two ago. So that helps. So I'm going to stop going through all of that because I think really we want to get to the highlight, and that is having our speaker. So it's important, though, that Joost has now moved to Perth since 2013, working at the University of Western Australia and focusing on the cancer immunology research. And in 2017 was awarded the NHMRC Excellence Award. And we're just so delighted that you're able to join us today. Dr. Joost Lesterhuis, please welcome him.
Thank you very much. I am an extremely quick speaker normally, so I'll try to slow down a bit. Thank you very much for having me speak here. My name is, I put it actually the pronunciation on the screen because I have a Dutch heritage, so my name is a bit funny, Joost Lesterhuis. I work in mesothelioma research, and I particularly focus on immunotherapy, so how to boost the immune system against cancer. And today I basically want to take you through some of that research that we are doing, and I'm not saying only I, but we at the National Centre of Asbestos-Related Diseases, which is at UWA. And I'll show you some examples of my colleagues and some of the examples that I'm working on. And I'll start with a bit of an overview of particularly what is mesothelioma, what are the type of symptoms, what is it, what we're talking about mesothelioma, that people, why do they get this cancer? And then I'll go a bit more into detail on the science. And I'll try not to make it too dry for you. And I have 45 slides for 45 minutes, so I hope that's sort of fitting. We'll see. So what is mesothelioma? Like every cancer, it always starts with a very normal cell. Normal cells, for some reason or another, and I'll get back to that in a few slides, turn into cancer cells. So what is the normal cell that mesothelioma derives from? We have this lining of our organs, our internal organs, which is called mesothelium. And basically, this it has to do with a sort of a little oily layer around our internal organs, our guts, our heart and our lungs. There is a bit of oil between these lungs and the surroundings to make these organs move freely. Now, in order for this oily fluid not to leak out, we need to have a cell layer that basically keeps that oil, that liquid in place. And that's mesothelium. It's only one single cell layer thick. So this is, in this case, what you're looking at here is a slide made by a pathologist of a lung. So you see the lung on this side, there's some air in here, and then you have the outer layer of the lung, there's a bit of fat, and on that there is one single cell layer, extremely thin. You wouldn't be able to see it with your eye. And that is these normal mesothelium cells. Now, for some reason or another, and I'll get back to that, these cells change their behavior. They start to behave in a disorderly fashion. They just suddenly start growing a lot faster than they should. They don't stick to their surroundings anymore. They don't stick to the fact that they should be only one cell layer thick. They actually become multiple cells thick, and they start to invade into the tissue that is lying underneath, such as the lung itself, or the heart, or the abdomen, or in rare cases, the testicles. So that is when mesothelioma arises. And what happens to patients in that case? So what you see here is basically mesothelioma as it grows. You have here the lung, and the heart with the aorta. We have the liver here, so it's basically like I'm standing here, cut through like this. Usually it starts either on the lung side or on the other side, on the side of the chest. These cells, they start to, as I just said, multiply, and you get a cancer. And in the case of mesothelioma, it's not a round tumor, but rather it's a rind-like growth. And that keeps growing, and then patients start to get symptoms. Because what you can imagine, if you have a thick layer around your lungs, you start to get problems with breathing. The other thing that can happen is that these cells start to make fluid, or they block the normal flow of the fluid, and people get fluids in their lung. And that, again, causes symptoms of shortness of breath. It can also grow into the chest wall, and then grow into nerves, and that causes pain. So those are typically the type of symptoms that people with mesothelioma would get: shortness of breath or pain. Sometimes, because these tumor cells need to divide a lot, they need a lot of energy. People feel tired. Those are typically the sort of symptoms that people can get. And then the most symptoms are caused by this local growth, the local process, but actually the mesothelioma can spread throughout the body. So these cells now suddenly, they don't behave at all anymore like normal cells. They actually don't stick to their place, but they move around through the body, for example, to lymph nodes or sometimes to the liver, etc. So that is sort of what mesothelioma is. Now what causes mesothelioma? And there's a very, very clear causative agent for this type of cancer, and that's asbestos. And that is actually the reason why here in Western Australia we have the highest incidence of mesothelioma per capita, as it's called, so relative to the number of people living in our state. And the reason that we have so many cases of mesothelioma has to do with this little town up north in the Pilbara, in the absolute beautiful gorges, close to Carigini. There's this town, Genome, where there was an asbestos mine. So asbestos is a mineral that you can just delve out of the ground like any iron ore or other minerals. And in the 1950s and 60s, there was a company up in Genome where a lot of people worked in the mines. As you can see here, this is a mine worker. They took out this asbestos and then they grinded it into little fibers, and that was then used as building construction material. So a lot of I'll show you that in the next slide, but you will find a lot of sheds around Perth or houses where they used this material. It was a perfect type of material from a construction point of view because it really is good against the heat. It's cheap, it's easy to make. So there were a lot of people in Genome who were exposed to a lot of these asbestos fibers. So either when they were working in the mine or when they were working in the mill, or sometimes even the people living in the town, there was a lot of tailings that were just complete 100% asbestos. So you see these children here, instead of playing in a sandpit, they're actually playing in asbestos. So what we have seen ever since, so this mine closed in the mid-1960s, and this is a bit of a complex graph, but what you basically see here, and this is the work from Professor Bill Musk, who really pioneered all this work in asbestos research here in WA. What we have seen is that these people who worked in Genome were really the first wave of patients with mesothelioma. And then the next wave we saw is this blue group. Those are the construction, the builders, people who cut the asbestos, put it into buildings, etc. And what we're seeing now is a third wave, and that's this one, where we see, so this is the relative number of patients per year, and this is sort of the area that we're talking about. It ends here in 28. I don't have newer data. I should ask Professor Bill Musk about an update. But what we see is this group is increasing, the third wave, and that is the people who are actually renovating their house, the do-it-yourselfers, who are now working in these sorts of houses, these old fibro houses that you see a lot around town, or under carpets that actually have asbestos in them, or like people getting rid of their asbestos fence. So that's the latest group that we see who have now are developing mesothelioma, and that's increasing. And then there is another group that's not in Australia, but actually around the world, it is perhaps of interest to know that, so the green countries are the countries where asbestos is banned. So Australia is one of them. We can't mine, we can't use asbestos anymore. But there are a lot of countries around the world where actually it's still allowed. And actually last week in the most prominent medical journal, New England Journal of Medicine, there was a whole piece about the current US government allowing again the use of asbestos, and it's still mined, for example, in the US. It's just been banned in Canada, I think about 3 years ago. So there's definitely a lot of people around the world who are still exposed to asbestos, albeit they are not always necessarily the blue asbestos, which is really the worst asbestos that we had here. But they are at risk of developing cancer as a consequence. So who gets mesothelioma? What we know is that there are people who have been exposed to large amounts of asbestos when they worked in Wittenum, but actually lived to a long age without developing mesothelioma. What we know is that it takes a long time before you're exposed to asbestos, before you then develop the mesothelioma. So typically 20, 30 years. The earliest cases, I think, are around the 15 years. But still, there are people who have been in Wittenum 40 years ago and they don't have mesothelioma. And then there are other people who have what we, at least based on their memory or their history, we can assess, have been exposed to only very small amounts of asbestos, if at all, and they do develop mesothelioma. So there seems to be some interaction, let's say, between your genetic makeup and the exposure to asbestos that could determine whether you will or will not get the mesothelioma, or the time that is needed for you to develop this mesothelioma. So one way that people at the National Center of Asbestos-Related Disease and other researchers, such as Bill Musk in Perth, have tried to assess that is by the fact that we know that there's a lot of people in the 1950s and 60s who worked in Wittenum. They were actually from different parts of the world, and particularly from different parts of Europe. So there were a lot of people from Italy, Croatia, Greece, and a lot of my fellow Dutchmen who actually worked in Wittenum. So one of the ways you can ask this question, is there an interaction between your genetic makeup and the exposure to asbestos in the development of mesothelioma, is by looking at all these people's genes and then see whether you can find a correlation between whether they had certain genes and whether they then developed mesothelioma. Now, we did that. But unfortunately, we can't find anything. And the reason is that these studies are extremely difficult. Why are they difficult? Is that people don't always behave like test animals. So in this case, what you will find is that some people would smoke and others would not. What you will also find is that the Dutchmen were actually taller than the Italians. And the consequence of that was that the Italians ended up in the mines and the Dutch people ended up in the mills. And the consequence of that is that these were exposed to a lot more asbestos than these. Now, did they get some other disease, for example, a heart attack, that could prevent them from being diagnosed with the mesothelioma later on? All these issues can play a role. So if you would really want to know whether there is an interaction between genes and the asbestos, you would need to expose everyone to the exact same amount of asbestos. And none of them should smoke, and you should all have sort of come from every single part of Europe so that you can test this. Now, obviously, that's not something we would ever want to consider. And this is where laboratory research comes in. Because, and this is not my work personally, this is, I should say, work from my colleague Dr. Scott Fisher and Professor Richard Lake, together with Professor Grant Morahan, who is actually a diabetes researcher at Perkins Institute. So what he did is, together with colleagues from around the world, developed mouse strains that are completely different. What I mean is that they are basically sort of the mouse variant of people coming from all over Europe, or actually from all over the world. So you have a lot of genetic diversity between these mice. And these mice, we can give the same amount of asbestos, and we can monitor them for a long time, and we can then actually try to find out whether there are certain genes that could prevent you from getting mesothelioma. Now, why would we want to do that? The reason for that is that if we know there are certain genes that could prevent you from getting mesothelioma, even though you have been exposed to asbestos, we might be able to develop drugs then following on from that we could prevent people from getting mesothelioma after they get asbestos. Now, this is what we aim to do. We're not there yet. So this is work that is currently ongoing, and I really hope that Dr. Scott Fisher can stand here in 3 years' time and tell you the results of these studies. But as it is, this work is very much ongoing at the moment. So this is really about why do people get mesothelioma. Now, what happens once you are diagnosed with mesothelioma? Most cancers, the first sort of treatment you will get is always surgery. But as I just told you, unlike a lot of other cancers, mesothelioma is not a clearly circular tumor. It's not a ball that you can take out, like breast cancer, for example. So it's a rind around the lung, and I've shown you here, this is a CT scan. So this is basically a patient, lying on his back with the feet facing you, and it's sort of sliced throughout the heart, or through the heart. That's sort of the level that we're at. So what you see here is the patient back, the spine. We have here the chest, the heart in the middle, and what you see here, this is a normal lung. This is what a normal lung looks like, and there's a one cellular layer of mesothelium, which you cannot see because it's too small. But what you can see here, this is mesothelioma. So as I said, it's like a rind. It goes around the lung, and the other thing is that it grows into the chest. So you can imagine that this causes quite a few symptoms in terms of pain and shortness. But you can also, shortness of breath. But you can also probably appreciate you can't take this out. This is not doable for a surgeon to take all this out, not even if you take the entire lung out. That's the problem with mesothelioma. As soon as it's diagnosed, this is often what you're talking about. So what we're then can do is give chemotherapy. And the goal of chemotherapy is particularly to reduce these symptoms. So, and it may come as a surprise to you, this is not necessarily what people have in mind with chemotherapy, but it's really to reduce and improve, or reduce symptoms and improve your quality of life. So if you have pain or shortness of breath, the aim of the chemotherapy is to reduce that. If patients respond well to the chemotherapy, it can prolong their survival. But it's a balance. There are side effects. It's not going to cure, and unfortunately this is the state as it is with mesothelioma. And the prognosis is unfortunately not what we want it to be. So this is why we're at the National Center of Asbestos-Related Diseases really aiming for developing new treatments that can actually attack this cancer. And that's basically what I wanted to talk about in the rest of the talk. And I'll start with some background on immunotherapy. I'll talk a bit about clinical trials, and then I'll end more about some lab research. So immunotherapy, you may have heard, you just alluded to it, Professor Michael Milward is doing very much work on that in the context of melanoma. So let me take one step back. What is immunotherapy? So basically our immune system is able to actually recognize in a very specific way cancer cells. So what happens is you have these killer T cells that roam through your body, and they're particularly good in killing virus-infected cells, but as a second job, they do a great job in getting rid of tumor cells. Unfortunately, tumors tend to outsmart us from time to time, and what happens is that tumor cells can put up a shield, and that's here in these little, I think they're green, but I'm slightly colorblind, so I might be wrong. It can shield itself from being recognized by T cells. And the consequence of that is that actually it escapes from the immune system. And it can grow. These killer T cells are not able to kill these cancer cells. Now, luckily, a few years ago, and I'll show you a few slides later, the people who actually discovered that, there were drugs identified that could actually take these shields away. basically making the cancer recognizable again by these killer T cells. And the consequence of that is then that these killer T cells can actually kill the cancer cells again. So it's basically inhibiting the inhibition. So it's taking away the brakes on the immune system, allowing these killer T cells to do their job properly against the cancer cells. And the consequences of that have been absolutely formidable in some cancers. And I'm going to show you this graph, which is a bit of a technical graph, but I'll guide you through. This is a so-called waterfall plot, because it has the shape of a waterfall. What you see here is the response of patients, in this case it's melanoma, to a certain therapy. In this case it's immunotherapy. We take a scan of the patient at start of the treatment, and we count all the total cancer burden. We put that in the number, so it's the diameter in centimeters, and that's the starting point. That's our point 0. If from there on the cancer decreases, then the line goes downwards. So 100% is here. That will be a 100% decrease of the cancer. So that will be a full cure. If the line goes upward, that will be an increase of the cancer following the therapy, meaning that the patient did not respond. So basically everything going down is good. Everything going up is not good. There is a bit of a gray area around the 10 to 20%, because we cannot be that certain in our measurements all the time. But basically what you can see here, every line is a patient. So what you can see in one blink of an eye is that about half of the patient respond really well to this therapy, and less than half don't respond to this therapy. The second thing you can see in this graph is there's quite a few here below this line of 30%, meaning there's a lot of patients that actually have a very substantial reduction in their cancer. And there's even a few that have 100% reduction. So they're basically cured. And this is absolutely astounding. And when we're talking about metastatic melanoma, that we now see these patients sometimes 10 years later, the first people who participated in these trials, and they're tumor-free. And that's absolutely fantastic. And the reason of that, that's probably the reason that it has had quite a bit of press in the last few years. Now, you might have come across all these articles in both the Australian as well as in the worldwide press. And last year, the Nobel Prize in Medicine was actually awarded to the two inventors, or two of the main inventors of these drugs. So Jim Allison and Tasuku Honjo. And it is because this is really, really impressive. But there is a but. And that big but is this. This is the same sort of graph for mesothelioma. And you can already see that it looks different from the previous graph. The first thing you see is that the number of patients who do not respond is bigger. There's a lot of patients who don't have much happening, and there's only very few who have this fantastic response. Now, I should say this is still quite a fantastic response. But it's only basically one patient that has the 80% regression, whereas what we saw in melanoma, it was quite better, a lot better. So for melanoma, this has been a game changer. A lot of other cancers, and mesothelioma is one of them, but brain cancer, pancreatic cancer, there's quite a few others, bowel cancer, they do not seem to respond at all to these immunotherapies. So the questions that we're trying to address here is first, why is this? Why do some patients and some cancers respond so really well to this type of therapy, while others do not? And the second question, perhaps even more important, how can we make this response better? How can we make all these patients, their tumors regress? So we want to see all the lines go down, basically, and preferably to the 100%. So I'll try to address these questions with some of the examples of research that we have been doing. And I'll start with, can we make these responses better? And this is actually work that we started quite a long time ago. So there was about, I guess, the last 30, 40 years, the idea has always been that chemotherapy is actually bad for your immune system, because we know that when patients get treatments with chemotherapy, they're more susceptible to bacterial infections. If you are receiving chemotherapy, you have to notify your doctor if you get a fever. So the idea had always been you cannot combine immunotherapy and chemotherapy. That turned out not to be quite that simple. When we looked into more detail, and this was, I did this during my PhD, and Professor Anna Nowak over here in Perth did it during her PhD. We sort of independently worked on the same thing. We found that actually chemotherapy has some beneficial effects on the immune system as well. So some of these killer T cells that I talked about before, actually they are better able to recognize tumors if those tumors are killed by chemotherapy. There are some other mechanisms. We actually found that some of those shields I talked to you about that are being put up by the cancer to protect themselves against the immune system are also being taken away by the chemotherapy. And quite a few other mechanisms we found that actually the response against tumors can be improved by some chemotherapies. So I was, this is in 2010, I was doing my PhD research in the Netherlands, and I looked around on international conferences, and in the literature I got myself a fellowship to go abroad for 2 years, and I was just actually telling that there were 3 groups in the world that were doing some absolutely cutting-edge research in this area. So one was Paris, the second one was in Baltimore, and the third one was in Perth. So in the end I chose to go to Perth, for family reasons, because I had small kids, and Baltimore was not the best place, I thought, to raise kids. And my wife, who is a doctor, didn't speak French, so that didn't work. And Perth, well, we looked it up on the map, and it looked pretty good. But it was the science. It was the science, really, that was cutting edge here. A group, Professor Anna Nowak and Professor Richard Lake in particular, together did a lot of research in this area. So I joined them in 2011, and we did some research. And I'm again showing you a bit of a dry graph, perhaps. This is called a survival curve, and this is laboratory animals. So what we have here, these are mice with mesothelioma. What you see on the y-axis is the survival, and what you see on the x-axis is the time. So these mice have mesothelioma, and we give them a treatment. So if we don't give them a treatment, these mice die pretty quickly. If we give them this immunotherapy that I just showed you, these wonderful results about in melanoma, we get similar to what we see in humans. With mesothelioma, we see that we get a few responses. You see that the curves moves a bit to the right, means they live a bit longer. And you see that about at 10%, the curve stays flat. That means we cured 10% of our mice. The same is true for chemotherapy. We see once in a while, we see a cure, but it's not much. However, when we put the both together, we suddenly see that we get a cure rate of about 60%. And we really see that it's very significantly delayed before these mice succumb to their mesothelioma. So based on these results and some other results that we had in the lab, we decided this should go into patients. If chemotherapy and immunotherapy work well together, we should try this in patients. So actually, it took us a little while. Professor Anna Nowak, who you see over here, who was also a Cancer Researcher of the Year last year from Cancer Council, she and I together designed a clinical trial in patients with mesothelioma, where we combined the standard chemotherapy that patients get already, and on top of that, we gave the immunotherapy. And we did this through the Australian Lung Cancer Trials Group. So that's really where clinicians work together around the country to get these sorts of trials up and running. So we actually got the pharmaceutical company on board. We had 12 centers in Australia, Melbourne, Sydney, Brisbane. Am I forgetting any? And it was led from Perth, where we treated 54 patients with mesothelioma with this combination. And it was really a very fast including trial, which is it's due to the fact that it was organized through this very professional collaborative, but it's also unfortunately because it's in Australia and we have a lot of mesothelioma. And these are the blink of an eye waterfall plot results. So what you see now is actually we have quite a lot of patients who do respond. Now I didn't show you this for chemotherapy only. I should say, if you treat patients with chemotherapy only, you also see quite a few lines going downward. The good thing, and that's encouraging to us, is that we see these lines going down quite substantially. So we see quite deep responses, whereas with the chemotherapy, this tends to be less deep. And the other thing that's noteworthy is that there's not too many patients that did not have any benefit at all. So this is really encouraging. It's not enough to change clinical practice. I'll get back to that in a minute. I just show you one example of the type of responses that we see in these patients. So it's a bit similar to the CT scan I showed you just now. So on the left, this is at the start of treatment. So the patient is lying on his back, facing with the feet towards you. This is the heart, and this is a normal lung. And what you see here is a big rind. That's the mesothelioma. And then after several courses of therapy, you see there's a tiny bit left here, but clearly most of the tumor that was here has disappeared. So that's really very encouraging results that we see from these sorts of studies. Now, if you know someone with mesothelioma, don't run to your oncologist just yet and say, I want this treatment, because this is what is called a phase 2 trial. So what that means that we try to see a signal of whether this has some effect in reducing the cancer. And we can answer that question with, yes, it has some effect. How much that effect is, whether people will live longer than if you were to treat them with chemotherapy, that's a next step. And that would be a phase 3 trial. So typically that's in several hundreds of patients where you let basically fate determine whether patients will get the standard therapy. So it's called randomized. So you draw what's the word I'm looking for? You have to basically draw well, anyway, it's completely I can't find a word. It's completely at random. You get determined whether you will be treated with chemotherapy or chemotherapy and immunotherapy. And that's the only way that we can really truthfully find out whether this is worth it, whether treating together chemotherapy and immunotherapy is better than chemotherapy alone. Now, that's not something we can do only in Perth. That's really has to be a international type trial. So my colleague, Professor Anna Nowak, is really working hard together with US colleagues to get that up and going. We're very positive about it, and I hope that soon we can get this study going. And if so, it will definitely be opened here in Perth. So going back to the question, so can we make responses better? I hope so. I hope we can make it better by giving chemotherapy. But can we try to address that other question? So why is it that some patients respond really well and others do not? And once we have that knowledge, can we use that to find out how we can make this response better? Just trying to... How much time do I have still? 8-9 minutes left. Yeah, okay. I might need to hurry up a bit. All right, because it does get a bit complex, I should say. So thinking about very complex, so what we have here, we have a cancer that has a lot of immune cells in it. We give a drug, and these immune cells are now suddenly able to recognize cancer cells and get rid of them in some patients and not in others. So we have a lot of different cell types talking to each other. We have a lot of different genes talking to each other, a lot of proteins. So how in this extremely complex mess, if you will, can you get an answer? And there's one person who could help us here, and it might surprise you who that person is. That person is actually Kevin Bacon. So some of you might be familiar with this concept. So there were Kevin Bacon is a Hollywood actor, famous from quite a few movies, and a bunch of college students in the 1990s were doing this game where they might have had a few beers, but they're watching this movie with Kevin Bacon, and they basically came up with this game where they could take any actor they knew, and within 6 steps they could link it back to Kevin Bacon. What I mean with that is that if an actor had played with another actor, and that actor had played with another actor, and that actor had played with Kevin Bacon, then there would be 3 steps. So as an example, George Clooney over here, I can't read the words very well, he played together with Julia Roberts in the movie... Can't read it? Might be Ocean's 11. Oh yeah, that's definitely true, yeah. And... And Julia Roberts played with Kevin Bacon in Flatliners. So that means there's 2 steps between George Clooney and Kevin Bacon. So these college students found that they could have any actor within 6 steps of Kevin Bacon. And actually, then scientists came on board. And there were 2 papers in 1999 in Science and in Nature. And in actual fact, and this is absolutely true, this paper is actually the highest cited paper amongst scientists in the decade following, that were basically about Kevin Bacon. So what they found is, so they looked into these networks. So they looked into these, so there's a database, IMDb I think it's called, where all the data from all actors and all their movies are basically put together, and you can actually do some math with it. And they found that the 6 degrees, the 6 steps, was actually a lot less. So any actor could be connected to any other actor within 3 steps. And the other thing they found is that there were a lot of actors that actually didn't play that much with others, so less than 10. So these were typically your, I guess, the one-off stars that played in one small movie, one small part. But there were a few actors who played with a huge amount of other actors. And these scientists then found that if you use network theory, you could explain what was going on here. So they could explain that you had a lot of actors here if you were to depict this as a network, where every actor is a node, and every co-acting is a line, an edge, then you would find that there is a few actors at the very center that played a huge amount of movies with a huge amount of actors and a very wide diversity of type of movies. So rom-coms, western, sci-fi, action movies. So they were the ones that were actually holding this network together. Because of them, everyone was connected to the others. And because of them, this 6 degrees, or actually 3 degrees of separation. If you take these guys out, it would suddenly increase dramatically. And what they also found is actually these principles, these mathematical principles underlying this actor network, also apply to all sorts of other systems, complex systems. Whether they were websites or airlines, genes, criminals working together, the power grid, or Swedish high school students, they all followed the exact same patterns. And this is really interesting because this means that you can actually look at these complex systems and apply mathematical methods and try to find what are the most important players in this. So it's a game when you're talking about actors, but when you're talking about cells, where you want to know what are the most important genes, you can actually perhaps tweak that system in your favor. So and the way to do that, so we can actually investigate thousands of genes in one sample. So what I'm showing you here, this is an example. So it's basically a switch. So we're measuring genes here in one sample. What we have here on the y-axis is a few hundred genes. If it's blue, it means the gene is switched off. If it's orange, it's switched on, and white is sort of in between. Now we can do that actually for all our genes. Thanks to the Human Genome Project, we know, have mapped all the genes, we know where they are, and we know we can measure whether they're switched on or off. And we can do this in thousands or tens or hundreds or thousands of samples at the same time. So what we did is actually measure all the genes in tumors from patients as well as mice with mesothelioma that responded or that did not respond to immunotherapy. So, and I'm just showing you sort of an example. So we have, and I'm just showing you a few, but basically we had 50 or 70 responding mice and 70 non-responding mice. And we had the same sort of data from patients. Now, you can imagine that if you want to know what genes are important here, you can't do that on the back of an envelope. You can't even do that in writing. It's impossible to do that using old-fashioned mathematical methods. This is really where computation comes in. And actually, sometimes even supercomputers. So for this sort of work, we have Palsy even. That's a supercomputer here in Perth, which is the most powerful computer on the southern hemisphere. Perhaps people don't know, but it's actually quite extraordinary here in Perth. And we can use this sort of methods, so computational methods, to ask the question. So what you can do is you can compare one gene, if it's being switched on, and you see another gene being switched on as well, every time you look at those genes across a lot of samples, then you can assume a link, and you can build a network like that. Now, you do this for one gene versus every other 30,000 gene. And you do that for every other 30,000 gene as well. So you can imagine that the amount of comparisons you're making here are just extraordinary. So that's really where these sorts of people, and there's particularly at Telethon Kids Institute, we have a wonderful collaboration with Timo Lasman, Melvin, Anthony Bosco, Emma de Jong, who do these sorts of analysis, where we actually compare thousands of genes with thousands of other genes. And what you can then do is basically try to identify those hubs, those key actors, if you will, that are actually driving the whole process. And you can actually let them, or try to then identify what are the most important regulators of a response. And what do I mean with that? So this is not, it's not about the details, but this is a picture of the network that we created when we compared responders and non-responders. And we found the Kevin Bacon in the middle, let's say. That's the gene that is really the one that was being switched on in these responding mice and not in the non-responding mice, as well as patients. So we do it in both at the same time. Now, the great thing by doing this in mice is that we can then actually test whether this is true. So what we did is we identified a drug, and it's an old-fashioned drug that has been around for a long time, whether that could switch on that particular set of genes in the middle. So it's one in particular, but there is actually a few more that are here in red and pink. When we did that, this is against a survival curve, and I say we, but it was actually Rachel Zemek who did that as part of her PhD. So again, if we don't treat mice with mesothelioma, they die relatively quickly. If we treat them with immunotherapy, we see a few that are being cured, like we see in humans, but most not. But when we give a drug for only 3 days that actually switches on this gene so that we can make this non-responder tumor into a responder one, we can now increase this, these mice, their response rate. So it's really an example of where you can try to, before you even start giving immunotherapy, we can sort of sensitize it to immunotherapy. So we can make this non-responsive tumor responsive. Now this is mice. It's not yet in humans. So this is really the next step that we need to make and see if we can get these sorts of drugs into clinical trials. We're not there yet. So I think this was my time, isn't it? So I think I'll skip the last one because that's a bit complicated. Well, let me show you this. I like complex. So what I just showed you is we looked at all the genes before we even treat people. But we can make our life even more complex. What happens in the cancer when we are treating it? Could it be that some decisions are being made while we treat patients? So could it be that if we give another drug on top of the immunotherapy, that we could even further improve it? And that's the sort of question we're trying to address here by not only looking at these complex networks before we treat, but actually during treating. So it would involve sampling a tumor with several day time points for 4 or 5 times. This is not something we can do in humans. So this is really where we went to mice, and we're basically now at the moment what you see here is the networks over time of a tumor that is on the left or that is not on the right responding to immunotherapy. And what we're doing now is churning through all this data and see if we can find genes that we can switch on or switch off in the right direction so that we can increase the response rate to therapy. And it might be that in the end we make our life very difficult, that we actually have to give some treatments at the same time or some sequentially. But we'll see. I don't know that yet. This is, I would say this is in the realms of conjecture. So this is really where we have to get some data going forward. So having much work is still to be done, but there's this wonderful YouTube movie, and I hope that someday it will come true, that if you see a movie of Kevin Bacon and you think of him and then actually you think he actually helped curing cancer. That would be wonderful, wouldn't it? So with that's what I wanted to say, so I'll end here. All the work I have been doing is it's in the Perkins building. So we're on the 5th floor, the National Center of Asbestos-Related Disease. We're part of UWA. And since about 9 months, I also have a group in Telephone Kids Institute where I focus particularly on sarcoma. I didn't show you anything on that today, but perhaps that's for 3 years' time. Thank you.
Good job. How important is amino acids for switching on and off genes? Thanks.
Well, amino acids, the regulation of genes is on many different levels. So there are certain specialized molecules that can bind to the DNA and switch them on or off. There are some molecules that can actually bind to whole bits of DNA and set about 100 or sometimes even more genes on and off. Amino acids, as part of the energy metabolism, and actually that's something I didn't discuss today that we're looking at in mesothelioma as well. There are big variabilities there. So there's, for example, mesothelioma uses amino acids, fats, glucose, all the energy storage that we have available to us. Cancer cells tend to use them differently than normal cells. And the reason for that is that you need a lot of building blocks for them to proliferate. So a normal cell would divide, let's say, every few months, but cancer cells can divide every few days. So you need a lot of building blocks there. And that's where amino acid metabolism comes in, and it can be quite different. Yeah. But it's not so much with the gene regulation as such.
With the oncology, does platinum still have a role, or has that gone off somewhere else?
Yeah, so platinum is a form of chemotherapy. So in the DREAM trial, for example, that we did, the standard chemotherapy was, in actual fact, platinum chemotherapy. And it is, at the moment, the standard therapy for mesothelioma. And it will be the comparison, if we can get this phase 3 trial off the ground, it will be the comparison. So this is the standard. Does adding in immunotherapy on top of that improve it? That's the answer that trials should give us if we can get that from the ground. So yes, platinum definitely has a role still.
And another one is, what is immunotherapy? What does that involve?
You mean practically?
Yeah, what do you do?
So it's basically an infusion. It's similar to the chemotherapy, where every 3 days, although we're more and more going towards longer schedules, where you might even get it every 4 weeks, you get an infusion at the ward or the outpatient clinic of about half an hour, I think. In terms of the chemotherapy, it has less side effects in general that we see than chemotherapy alone. And when we add it on top of the chemotherapy, we don't, as far as we saw in those 54 patients, it doesn't add extra side effects.
The infusion is, what is that? It's not a mint tea? No.
Sorry?
What is the infusion? What does that consist of?
Sorry. So you go to the outpatient clinic, you get an IV drip. Yeah. I think you get one shot of a bit of saline through it, so that you know that it works well. And then in half an hour, you get this bag of the immunotherapy. Yeah. And then after that, you're fine. What is that?
What's in the bag?
In the bag is the drug. So the drug is actually an antibody. So, meaning that it's not dissimilar to antibodies that we make ourselves against viruses. But the thing about antibodies that we can use as drugs, so they're made in large cell cultures, and they're made against one specific target. So in this case, not a virus, but against those shields that I showed you. So the shields that shield away the cancer cells from the immune system. By the antibody blocking that shield, it basically sort of pulls us away, and the immune cell can do its work again. So it is now able to recognize the cancer cells. With that in mind, you might also appreciate that there are side effects that are associated with this, are immune side effects. So we're not doing this in a specific manner, right? So we're taking these shields away. These shields are not only on cancer cells, they're sometimes also on healthy cells. What we sometimes see, particularly in patients who respond well, is so-called autoimmunity. So the immune system actually attacks some healthy cells. So what that could be is, for example, the thyroid is being attacked by immune cells, and you get hypothyroidism, which is a treatable disease in itself, but that is a side effect that we, from time to time, see.
Mm-hmm. Thank you.
I'll wait till you have the microphone.
Who was it? I'm just wondering where you harvest the cells for the cell cultures.
So in this case, we're not treating with cells, we're treating with the antibody. And this antibody is being made by pharmaceutical companies by these engineered cells. Yeah, but we're not treating patients with cells. We're just treating with the antibody. It's basically a drug. Yeah, it's a protein drug. Yeah.
Probably time for one more. Anyone's got something? I think please join me once again in thanking Dr. Lesterhuis.
Thank you.
I don't think we've ever had a Cancer Update speaker talk about Kevin Bacon before. So that was very entertaining, so thank you very much. I do just have a few more announcements, and that is please don't forget to fill in your evaluation form. It really does help us, and if you've got any ideas on what you would like to hear in the future, that can really help us as well with planning other update topics. If you would like to hear about upcoming lectures, we can also add you to our email list. If you have email, it's the easiest way for us to get in touch with you. So please complete your details at the bottom of the evaluation form, and we'll separate those so that your evaluation's anonymous if you want to. Also a reminder that if you think someone you know would like to see or hear today's presentation, please do let them know. It will be on our website. We do promote it on our Facebook and Twitter pages as well, but do please let anyone you think might like to know about that. And also, just for future cancer updates, maybe just pop onto our website if you're not on email so that you can see what ones might be coming up in the future. But once again, please join me in a really fantastic presentation on mesothelioma.